my name is Linda Rost, and I'm a science teacher at Baker High School in Baker, Montana. I teach 10th grade biology and 11th and 12th grade biology too, 11th and 12th grade chemistry, and then I also teach a science research class. And I will be presenting on how I have been implementing the next generation science standards in my classroom, specifically my 10th grade biology classroom is the example I used, and how I used Vernier technology to do this. This is a presentation documenting my transformation from a one-dimensional teacher into a three-dimensional teacher in terms of the next generation science standards. I've been involved in a program for the last 2 years that's helped me to implement these in my classroom across the state of Montana. And what this one dimension and three dimension refers to are the three parts to the next generation science standards. The typical part that we normally think of are the disciplinary core ideas, and those are the typical content standards that we've had before in standards. The two pieces that next generation science standards added were the cross-cutting concepts and the science and engineering practices. The cross-cutting concepts include things that you will see across all fields of science and even other fields like patterns, cause and effect, systems and system models, structure and function, things like that. And then the science and engineering practices include a list of 8 items practices that scientists do and that we should be teaching our students to do. So that's the focus, and they include things like asking questions, developing and using models, planning and carrying out investigations, and other things. And these two parts, the practices and the cross-cutting concepts, those were my focus this year as I implemented these standards. So here's another look at the 3 dimensions. Again, you have the content, the practices, and the cross-cutting concepts. This is a graphic from concord.org, and they have resources that help teachers implement the standards, and it's a neat way to see how they all intertwine together. And for my example here, the content that I used was photosynthesis, and this is something that I really struggled with my 10th grade biology class. I struggled with finding a way to teach them differently, and what I found was a lot of times my students were leaving my class not knowing any more than they did coming in about photosynthesis. So that was really my challenge this year. I also challenged myself with adding the engineering practices into my teaching. I had a lot of students who had a lot of interest in engineering, so I really struggled with integrating engineering into biology, so I really focused on that practice too. So before, my one-dimensional lessons with photosynthesis would include maybe worksheets, some labeled diagrams, maybe things like this. We would study this. This would be in the book. And we'd have this diagram showing photosynthesis and cellular respiration and the contrast. And we would definitely focus on the equation, make sure everybody understood that. Then last year, when I integrated 2 dimensions into my teaching, we also did the worksheets and we labeled diagrams and things like that, but I integrated more practices, so I had them construct explanations by writing about the process in different ways. They also had to design and plan investigations. They had to design their own experiments, so there was inquiry there. And then we also modeled the process. We acted it out. We did things like that, but it was still not very rich. There was still something missing. So I wanted to do more, especially with this unit. So this year I integrated the 3rd dimension into my classroom, and that is the cross-cutting concept. The one I chose to focus on is what we call energy and matter, flows, cycles, and conservation. I also could have focused on structure and function, but I chose to focus on energy and matter, and that formed my lessons completely differently than if I would have focused on structure and function of the chloroplast. So the lesson that I designed for this cross-cutting concept with the content of photosynthesis and using the practice of engineering, I designed a solar cell challenge where students made a model of a chloroplast, a working model, and this model could collect light and it converted it into energy. And to make this model of a chloroplast, we actually extracted plant pigments. And their challenge then was to extract the best pigments to optimize the amount of light absorbed by the model chloroplast. And so there I emphasized that I also was integrating engineering. So their goal was to engineer the plant pigments. This is a picture of what the solar cell looks like. And up here, I purchased this from Arbor Sci. That's where I purchased the kit from. And it comes with these glass electrodes, 2 of them, and you put you add this white material to it, and then you dye this with the plant pigment, and you put them together. You add some iodine and you connect it up to a multimeter, and then you can collect data on your model chloroplast. This is a diagram from the information of what it looks like, so you can see the dyes adhere to this electrode here. You have to use certain dyes and certain plant pigments to make this happen. So the first step here was they had to prepare the glass slide, and then the main step was design plant pigments to capture the full spectrum of light, and then they soaked their glass slides in the pigment that they designed, and then finally they tested their solar cells. But the focus was, again, designing the plant pigments. Some of the prior knowledge they had to have for this is they had to know basic knowledge about photosynthesis, and I did this by teaching one-dimensional and two-dimensional lessons, so I'm still not completely converted. I don't know if that's possible for me because we still had to look at the content in kind of an efficient way, but students knew that at the end of doing this unit, they would be doing the solar cell challenge. So they knew that they had to learn this information in order to be successful with the solar cell challenge. And I do a lot of lessons like that so that they have a goal in mind and they're going to use the knowledge somehow. They also specifically need to know that plants take in light energy and convert it into chemical energy in a two-step process. They had to know about the visible light spectrum. They had to know that all the light colors add up to make white light. And then this is kind of a joke in our classroom now. Plants are green because they aren't green. So because plants absorb blue and red, they reflect green. That's why they look green, but they don't absorb green. In order to do this experiment, I also added the use of the SpectraVis Plus, and that's a product that's made by Vernier. And the SpectraVis Plus, if you put the pigment of the plant inside this cuvette and put it in the machine, this is the output you get on the Logger Pro program. And so it shows what wavelengths of light are absorbed at what levels. And you can clearly see here the peaks for chlorophylls A and B. So this would be your typical green plant. And then this is an example of what anthocyanin would look like. So you have a peak more in the green area. So if students were to add these two pigments together, their goal was to get all of the light colors absorbed to maximize the amount of wavelengths absorbed by their solar cell. So there's the device that I used, and this measured light absorbance, and this allowed this process to be really iterative for the students. They were able to make plant pigments again and again and keep testing them to see what the output would be. Now I'll show a video of the students working and designing their plant pigments, and then I'll wrap it up at the end.
Raspberry.
What?
Oh, keep talking. No, we should switch to raspberry because these two both have more color. It's going to take away the color from that one.
Okay. Guys, we should just put all the colors in there.
We're gonna Google what's best.
Oh, I don't know. It really hasn't been studied, honestly. You test it.
We'll try it. This will kind of make it a greener, lighter color. Kind of sort of.
How about you just pour some of the liquid? No, don't throw that in there. Just pour the liquid in there.
Putting some mix of everything. Holy crap! Yeah. I'm pretty sure most of us have a mix of everything.
What do you have?
I have like mine's lots of blackberries, some blueberries, and then algae and water.
What do you have?
Just blackberries.
And you're just doing raspberries?
Raspberries and blueberries.
Raspberries and blueberries. And I did one of everything.
I feel like blackberries are best because like
Yeah,
they have lots of
really
colors
rich
and they have like and they're like light but also dark. Are you videoing this?
We're trying to get as many colors in it as possible so that it turns black eventually.
How long do we do it in the mixture?
Or as close to black as we can.
Arrows. Okay, so what are you seeing there?
It absorbs a lot of blues and
What do you want it to absorb more of?
the greens.
Can I borrow one of
Yellows.
these?
The yellows.
And
And red.
the red. So what are you going to add to make it absorb more red?
So you want this, right?
Raspberries because They absorb.
No, they reflect.
So blackberries.
You guys, maybe we should try.
But also, if you add more green, they're going to absorb more red because chlorophyll is green and it absorbs a lot of red.
Some more algae.
And we're going to go to the greenhouse pretty soon to get more of your green.
Fudge.
So you want to see more in the red too.
Yeah, so
So what could you add to get it more in the red?
probably
Don't go too deep. Just like try to get some off.
I got no idea.
What color would you put in to get more red absorbed? You'd have to put the opposite of red.
So
opposite.
Don't go in really deep.
we don't have any purple, do we?
Well, the blueberries and stuff. What did you put in there?
Blueberries, raspberries, and also blackberries.
Okay, if you put green, green is also going to absorb red.
Okay,
Okay,
so
so we
So that's,
need,
yeah, so that's why we're going to the greenhouse.
Alright,
so that's what we were missing.
Get a little bit of everything. These things. This is dark. Take a fern, strip this off.
So you might be,
Try to get more.
yeah, there's a whole bunch down in the bottom. It's all on the bottom there.
Oh, there's a big one.
If it has pink colors on it, then.
Your side. Those might be kind of neat to look at.
Because I need to usually means unstable.
Here, I got one more.
Oh, Lord. Stubborn the forest.
Oh. Do we want like a separate scratchy thing for these?
You could. And you could test it separately. You could test each leaf separately to find which one has the best, you know.
I think we're good.
Okay.
Alright, we ready?
So this is a picture showing how they dye the solar cells, the electrodes. They just dip it in their plant pigment that they designed once they finished engineering it. And then this is what it looks like to construct the solar cells. They put two electrodes together and clamp them with binder clips, and then they hook them up to a multimeter to test the volts and amps. And the goal was obviously to get the solar cell with the most voltage and amperage, or one or the other. And to do this, they really focused on getting plant pigments to absorb most of the visible light spectrum. So in that way, they were really able to understand how plant leaves work better and how chloroplasts work better. And then they had questions afterwards, like why aren't plant leaves black or dark, really a dark color, because that's what pigment would absorb the most colors. So we talked about problems with that and cell damage if they absorb too much energy, and so there had to be a balance. So that actually kind of led us into our next unit, which was on cancer and mutations. So this is, again, a contrast between what I used to do, and some of this I still did at the beginning for prior knowledge. This is a notebook example. We do science notebooks, so this is some of their notes from what they needed for prior knowledge. So we showed the light and dark cycle and what goes on during each. Here's another example of a science notebook on photosynthesis and chemical energy. So that's what we did before we did this challenge. And then this is what the actual write-up looked like from the challenge. So I had them do a purpose hypothesis, and we always do our independent, dependent variables, our control and constants. And then this is an example of a graph. Every time they engineered a pigment, they had to test it with the Spectrovis Plus, and they printed off the graph and put it in their notebooks every time they made a new pigment, so they could keep track of their improvements. And so that's an example of what the graph would look like in their notebooks, and then they had to do a write-up to show that they understood it. This is what their procedures looked like. I don't give handouts anymore for procedures. I demonstrated the procedures, and they had to write down step by step what to do, and they had to use those procedures to do the experiment. So they had to rely on their procedural writing to do that. Here's just another notebook example of mitosis that we did, and some cancer discourse. And this is a lesson inventory that I use for the next generation science standards. And on This side over here is just a big idea of the lesson. It says design solar cells to model energy flow in chloroplasts, and my disciplinary core idea was LS1.C, and that was organization for matter and energy flow in organisms. Right here, these are the cross-cutting concepts, and again, I used energy and matter in systems. And for the science and engineering practices, I used developing and using models and constructing explanations and solutions with a focus on engineering. For my performance tasks, I just have a list here of example performance tasks that could be done while implementing the next generation science standards. So they used lab design and analysis. So students conducted authentic investigations and asked their own questions to test. And they also developed models. So the model was the model of a chloroplast. Over here, I have how could engineering be implemented into this lesson, and I focused on biological and chemical engineering. So the engineering focused on designing plant pigments to optimize visible light absorbance. And the next question is how can student-led investigations be implemented in this lesson? So I said as students designed plant pigments, they tested light absorbance in a spectrometer, which is Spectrovis Plus by Vernier, and then they improved their mixture and continued testing it. Each group shows different factors to vary, and as it showed in the video, quite a few groups actually isolated the pigments from different plants and tested them separately, and then wanted to see if they could add them together to see if it had additive properties of the light absorbance, and a lot of them found they didn't. So let's say one plant did really well absorbing in the purple and another one did really well absorbing in the red. If they added those together, they didn't see those properties added together, which was really interesting. So some of the key questions and student understandings to assess, how I assessed them, they needed to know that plants absorbed light and converted it into energy, which we modeled with the solar cells. They needed to know that plants do not absorb the green light of the visible light spectrum, and also that technology can be modeled after living things. A lot of times we study living things and then we design technology based on those living things. So to sum everything up, this was a really successful engineering activity that we did, and it would not have been possible to add the engineering piece if we would not have had access to the Spectrovis Plus, which I obtained through a grant, because students would not have been able to test their pigments, and they would not have had such a good understanding of plant pigments and how plants absorb light energy. So that was one way this year that I integrated the next generation science standards in engineering using Vernier technology. I use Vernier technology a lot in my classroom, and I'm always, every year I try to buy a few more probes so that my students have access to that, but it seems like we never have enough. And I think the idea of for students to use quantitative data and to know that there are all this variety of probes out there for them to collect quantitative data on anything they can dream of is a really effective way to teach science, because they're doing it the way that scientists do science, which is the focus of the next generation science standards.